In this final episode of Epigenetics Explained, I hope to shed some light on the therapeutic potential of epigenetic drugs. So, what is epigenetic therapy? It aims to reverse epimutations which lead to cancer, as well as other diseases. Many of the most clinically advanced approved epigenetic drugs are targeted towards blood cancers such as leukemias and lymphomas. Developing drugs targeted towards solid tumors is much more challenging. Epigenetic therapy can be combined with immunotherapy, chemotherapy, and radiotherapy to maximize its effectiveness. Let's discuss the different types of epigenetic drugs available. I will be focusing specifically on DNMT and HDAC inhibitors. DNMT inhibitors contain cytotoxic agents that inhibit specific DNMTs. These DNMTs regulate genes involved in apoptosis and differentiation. Currently, there are 2 DNMT inhibitors approved by the FDA. The first drug is called Vidaza. This drug was approved in 2004 and has a response rate of 20 to 60%. This refers to the percentage of patients whose cancer shrinks after treatment. The second drug is called decitabine. This drug was approved in 2006 and has a response rate of 40%. Both drugs target myelodysplastic syndromes. These are diseases that affect the production of blood cells in the bone marrow. However, there are a few issues associated with these drugs. One is that they reduce the overall production of blood cells. And another is that they are unstable in aqueous solutions. Now, these are solutions in which water is the solvent. This makes them hard to transport and store. The other type of epigenetic drug is HDAC inhibitors. These impair the ability of histone deacetylases to recognize substrates, which are the substances they act upon. This helps to restore normal histone acetylation levels and reactivate cellular pathways that promote apoptosis, cell cycle arrest, which is a pause in the cell cycle, and differentiation. There are currently 2 HDAC inhibitors approved by the FDA for treating T-cell cutaneous lymphoma and 2 other drugs in clinical trial. In the future, epigenetic drugs could be developed to target histone methyltransferases, histone demethylases, and histone readers. But let's take a step back to understand how epigenetic drugs are developed. This is a rigorous process full of regulatory hurdles which takes years to complete. The first stage is a drug screen. During this stage, drugs are tested on cells for effectiveness and functionality. If the drugs have an inhibitory effect on the growth and proliferation of cells, they are deemed promising and move on to the next stage, which is epigenetic profiling. This involves identifying and assessing the effect of a drug on the cell's epigenetic profile. The next stage is preclinical testing. This involves testing drugs on mouse models for toxicity. The first 3 drugs are all performed in a laboratory setting. The final main stage of drug development is clinical trials. This is where drugs are tested on humans. It is divided into 3 phases to assess for dosage and safety. The final stage is approval by the FDA or other regulatory authorities. This isn't the only barrier between the lab and the clinic. Other issues to consider include concerns that targeted genes are pleiotropic. This describes a gene that produces more than one phenotypic trait. This poses an issue because interference with pleiotropic genes could cause reactivation of potentially harmful phenotypic traits, ones which could cause disease. Luckily, there hasn't been any research to back this up yet, but the issue could emerge. Another issue is that patients could develop spontaneous or acquired resistance to drugs, which dampens their effectiveness. Regardless, the field of epigenetic therapy is growing rapidly and could provide yet another alternative for cancer treatment. Well, that wraps up the final episode of Epigenetics Explained. This project is unlike any other I've done, and I'm so glad I can share it with you. Thank you so much for watching. That's all from me.